So I am Gurudev Singh, retired scientist from the Institute of Forest Genetics and Tree Breeding, Coimbatore. So after retirement, I have taught the tree improvement subject to the postgraduate students and also the PhD scholars at the College of Forestry, Pannampet, Kodagu. So today I'm going to talk on the breeding cycle. So breeding cycle is if you understand breeding cycle, you are understanding the heart of the tree improvement program. Very important topic. And if you understand breeding cycle, then it is you will understanding the tree improvement, the heart of the tree improvement. So we say that the tree improvement is based on the mass selection and recurrent selection. So what is meant by mass selection? The mass selection means, unlike agriculture crops, we breed here group of trees. That means we select many plus trees and we deploy them in a seed orchard. So this is the mass selection, we allow them to mate. So many plus trees are assembled in seed orchard and allow them to mate. So that's why it is, we call it as a mass selection. And second thing is we call it as a recurrent selection because we have got different cycles of selections. It is not that one time we select the trees and we deploy it and it is over. So we allow them to breed and we again select the best individuals from those progenies and use it for breeding the next cycle. So you must have heard the first generation seed orchard, second generation seed orchards, third generation seed orchards, and such a cycle we call it as a recurrent selections. So we select from almost the same base population by allowing the many recombinants. So that's why we will call it as recurrent selection. So it is mass selection and recurrent selection. And this is a chart of tree improvement in a concise manner. I will try to explain. So in tree improvement, what we do is we select the plus trees. So what is the plus trees? Plus trees are nothing but outstanding individuals which we select for the given characteristic features. So if we are selecting a timber species like teak, we give importance for the total height of the trees, the clear bole height, the girth of the trees. If it is for fruit yielding trees, we give emphasis on the size of the crown, the fruit yield of the trees, and such aspects. So from where we can select these plus trees? We will select these plus trees either from the natural forests or from the plantations. So many plus trees are selected and based on the various parameters, and these selected plus trees are deployed in the seed orchards. Basically there are two types of seed orchards. One is clonal seed orchard, the other one is seedling seed orchard. In the clonal seed orchard, what you do is from the selected plus trees we collect the vegetative material either in the form of cuttings or grafts, and we allow them to root and we will plant them in an orchard. So since we are using the vegetative material, we call it as a clonal seed orchard. So many of these plus trees are planted in this clonal seed orchard in a particular design. So the other type of seed orchard is a seedling seed orchard. Here what happens, from the selected plus trees, we collect the seeds, then we raise the seedlings, we plant the seedlings in the orchard in a particular design. We call it as a seedling seed orchard. So when your vegetative material we are using for establishment of orchard, we call it as a clonal seed orchard. Whereas when you are using the seedlings for planting, we will call it as a seedling seed orchard. So once these seed orchards have been established, we allow the pests to mate among themselves and produce good quality seeds, and these seeds are used for raising the plantations either by the private industry or by the forest department. So this is the process: select the pests, deploy in the orchards, collect the seeds for raising the plantations. When we are selecting the pests, we are selecting the pests based on their external feature. So that is, we will call it as a phenotypic selection. We will not be knowing whether they are genetically also superior or not. So in order to understand whether they are genetically superior or not, concurrently, when you are establishing the seed orchard, simultaneously, we will establish a progeny trial. So the progeny trial is something like a test plot, and the data what we generate from the progeny trial, we use it for culling the inferior trees from the clonal seed orchard as well as seedling seed orchard. So the progeny trials are important to know the genetic worthiness of the parent trees. And when we establish this orchard, we call it a first generation seed orchard. And when you do a culling, we call it as a 1.5 generation seed orchards. The same pestis we can produce, we can use it for production of the artificial hybrids. So we will have two individual trees, and we allow them to mate. We do a control crosses and produce the artificial hybrids. So these artificial hybrids can be within the species or between the species, intraspecific hybrids or interspecific hybrids. And these hybrids as well as the pestis vegetatively we will propagate, and we will maintain them in a clone bank, and this clone bank will serve as the base for establishment of the clonal seed orchard. And this clone bank nowadays are being used for establishment of the clonal plantations. So clone bank is something like a germplasm assemblage of the pestis, and from this clone bank we select the best clones and use it for large scale clonal production and use them in a clonal plantations. And if you see in this chart, I have also marked the seed production areas as part of the tree improvement. So seed production areas are also meant for seed production, and it is not that in the seed production areas what we do is we select the best plantation, and from this best plantation we remove the inferior trees and we maintain average and above average trees, and after culling we will collect the seeds from this seed production areas. So seed production areas can be established in 2 years, 1 or 2 years time, whereas the selection of pestis and establishment of orchard and collecting seeds will take about 10 to 15 years depending upon the species. And if you understand this, the whole chart it gives the idea about the heart of the tree improvement program. So next we'll talk about the breeding cycle. So breeding cycle, it starts with what we call it is the base population. So base population means it is the selections from the natural forest areas or the plantations what you have selected. There is large number of trees we will be using it as the base populations. So from this base population what we do is we select a good number of trees, we call it as a selected population, and this selection we will do it based on the progeny test. We do the genetic test, then we select. So for example, if you have got here base population in lakhs of trees, we may use some thousands of trees in a selected population. So from the selected population we establish a breeding population. So the breeding population is the core population which comes from the selected population. So this may be breeding population may be few hundreds of trees, about 300, 400 trees, where the selected population can be few thousand trees. So in between, when the forest department is in a requirement of the seeds, what we do is, meantime, we'll establish what is known as propagation population, and this we will also call it as the seed orchards. And from these seed orchards, we will collect the seeds and use it for the plantation activities. So the breeding population, we allow them to mate amongst us, and again, after intermating, we use it for next generation breeding. So I have shown here it as a base population. It is not the same base, it is some other base. It becomes a base for next generation breeding. So don't get confused that it is coming back to the base population. No. So it is a different base population which is used for breeding for the next generation activities. And when you do a continuous breeding for about 3-4 generation, the genetic base becomes very narrow. Then that time we will add an external population. We will call it as a infusion population. So this can come from outside, from another breeding program institute, and that will help us in widening the genetic base. And you will understand now better with this breeding cycle. So a natural population which is there, from there we do the selection and we establish the base population. We then go for selected population. From selected population we go to the breeding population. So this may be large number of trees, here few thousand trees, and here it is few hundred trees. So this breeding population, we allow them to mate and collect the seeds from here. Please remember, we are collecting the seeds from these selected trees and we use it as a base population for the next generation breeding. So here it is, we call it as this one as the first generation breeding, which I have represented with the green circle. And the seed orchard what we are establishing for commercial purpose, that is the propagation population, and we call it as a first generation seed orchard. So, and from the first generation seed orchard, the breeding population, we will establish a second generation base population. Again it will continue here, selected population, and from the selected population few will become the breeding population, and that will go further for the next third generation of breeding. And in the second generation, when you establish the production population, a commercial seed orchard, we call it as a second generation seed orchard. And likewise it will continue in the third generation, base population, selected population, and breeding population. Like that it will continue. So it may take decades, 1 decade maybe to complete one generation, another decade for the second generation, likewise for the third generation. So here if you understand this concept of the breeding cycle, so first generation, first breeding cycle, second breeding cycle, third breeding cycle, this is the first generation seed orchard, this is the second generation seed orchard, and this is the third generation seed orchards. So if you understand this concept, this becomes a heart of the tree improvement program. And as you go on, we are going to see other aspects of the tree improvement. So the tree improvement, maybe in extra class I will handle about the variation, selection, again seed orchard, seed production as provenance resource, control classes, clonal technology, design of experiment, genetic parameters, ANOVA, and seed certification. So I'll stop here, and next my presentation will be on the mostly variation. Thank you.